this is a schematic representation of the intestinal barrier, which is more as functional, is both a structural and a functional concept. And the intestinal barrier, the main function is the intestinal barrier is essential to maintain the symbiotic relationship between the bacterial communities of the intestine and the host, avoiding the penetration of the commensal or pathogenic bacterial to the host tissues. And to accomplish these functions, the intestinal barrier has different feedback immune system loops, such as epithelial toll-like receptors and dendritic cells, which trigger the production of antimicrobial peptides and immunoglobulin. And another important concept is that to keep tolerance to the dense bacterial communities of the intestinal barrier has developed two strategies. A stratification of these bacterial communities within the mucus layer and compartmentalization of the intestinal immune responses, mainly through the activation and the presence and the recruitment of regulatory T lymphocytes. And these three levels, you see schematically that there are three levels of defense in the intestinal barrier. The three are disrupted in cirrhosis. And the high rate of bacterial translocation observed in cirrhosis is due to the simultaneous disruption, the simultaneous failure of these three levels of defense in the intestinal barrier. Changes in microflora, alteration of the epithelial integrity, and impairment in intestinal immunity. And in this talk, I will briefly review all these abnormalities with a special emphasis in changes in the intestinal immune system. And the previous speakers have already shown this slide and the results and the different aspects of the qualitative and quantitative changes in intestinal microbiota that are observed in cirrhosis. To make short this long history, there are qualitative changes in microbiota in cirrhosis. There is a relative decrease in potentially autochthonous taxa such as Clostridiales, Lactospiraceae, Ruminococcaceae, and a simultaneous relative increase in potentially pathogenic taxa such as Enterococcaceae, Streptococcaceae, and Enterobacteriaceae. And in this recent paper in Nature, it has been observed in this abundance-based species network, which is very hard to reach. You see an increased number of this almost blue color, that in the bowel of patients with cirrhosis there is an enrichment in these patients with species of oral origin such as Taxa veillonella and Streptococcus, indicating that the oral commensals invade the gut in the patient with cirrhosis. And regarding the changes in microbiota in cirrhosis, it is important another concept that has previously shown that these abnormalities parallel the severity of the disease. In this slide, you see that these changes correlate, for instance, the reduction in Lactospiraceae and the increase in Streptococcaceae parallel changes in Child-Pugh score. And regarding the cirrhosis dysbiosis ratio, this relationship between the good and the bad fellas, the anti-inflammatory fellas in the bowel and the bad fellas such as the Enterobacteriaceae, this ratio progressively decreases as the cirrhosis progresses. But we also know that in cirrhosis there are not only qualitative but also quantitative changes in the microbiota, and this is intestinal bacterial overgrowth that since many years ago we know that they are linked, associated to most episodes of bacterial translocation. And to briefly summarize the potential reasons for these abnormalities, these quantitative changes, I would highlight the presence of intestinal hypomotility in cirrhosis, the impairment in the innate mechanisms of defense in the intestine. I will tell you something else later on, and the impairment in bile flow and bile composition that we shouldn't forget. Regarding the second level of defense, the integrity of the intestinal epithelium, it is well known that in cirrhosis there are structural and functional changes in the intestinal mucosa. With conventional microscopy, it is known that in the ileal mucosa and in the colon mucosa of patients and experimental models of cirrhosis, there is congestion and edema, and what is more important, alteration, abnormalities in the tight junction proteins, the mechanical seal between the epithelial cells. And these changes, this alteration in the expression of tight junction proteins have been demonstrated in patients with cirrhosis in duodenal biopsies, not only in the compensated ascitic patient, but also in compensated patient, indicating that it is an early event in the evolution of cirrhosis. We have been able to show it, as we presented in the last EASL meeting, that also in carbon tetrachloride cirrhotic rat with cirrhosis and ascites, there is altered expression of tight and adherent junction proteins such as CO1 and E-caterin. And what the direct consequence of these abnormalities in the integrity of the epithelial barrier are an increased permeability to macromolecules that have been observed using DTPA and different sugar probes, and have been observed both in experimental models and in patients with cirrhosis, mainly those with ascites. But here I would like to raise a word of caution, because these sugar probes measure paracellular permeability. And in other models, it is known that translocation of whole living bacteria mainly occurs through the transcellular route, a possibility that has not yet been explored in cirrhosis. And going forward, we are in the third level of defense of the intestinal barrier, which is the subepithelial layer, where important interactions take place between the immune system and the content, the luminal content. This is the bacteria. And you should think that this intestinal immune system is subjected to the increased enteric bacterial load of the experimental model and the patient with cirrhosis. So it is not unlikely, it is not rare to find the presence of inflammation in ileal lamina propria of the of the experimental model of cirrhosis, as we observe in this slide. And this inflammatory infiltrate predominate T cells, T lymphocytes. The analysis of this inflammatory infiltrate show us that there is an increased number of activated Th cells, effector memory Th cells, and also TC cells, which are represented in the blue bars. And they are activated and oriented to produce pro-inflammatory cytokines such as interferon gamma, and in the final analysis of the ileal mucosa, there is an increased expression of these pro-inflammatory cytokines. And what is also important, that there is the increased enteric bacterial load is inducing or is participating in the induction of these pro-inflammatory changes, this inflammation in the small bowel mucosa of the cirrhotic rats. Since bowel decontamination with non-absorbable antibiotics normalizes this state of inflammation. You should also remind, I've previously commented on, that the presence of pro-inflammatory cytokines is a reason, is a well-known player for disrupt the tight junctions through the MLCK activation, further increasing intestinal permeability. And a situation that we do not understand yet very well, but we have some clues, as is shown in this slide, it's why this persistently and increased enteric bacterial load does not lead to a situation similar to the one you can find in inflammatory bowel disease. And we think that the reason is that in cirrhosis, the recruitment of T-regulatory FOXP3 cells is preserved and keeps the inflammation, the intestinal inflammation, contain the inflammation in a reasonable grade. And so the first alteration I have just shown is that the small bowel in cirrhosis shows inflammation. And the persistent antigenic, and in other matter of things, the persistent antigenic bacterial pressure present in cirrhosis also shapes the function and the functional behavior of other immune system cells, intestinal immune system cells, such as dendritic cells. And if we analyze the production of TNF and the phagocytic ability of the dendritic cells, we observe that this constant antigenic bacterial pressure induces changes that range from appropriate activation and increased function to relative tolerance, to relative exhaustion. And what is again more important, the link between this relative immune deficiency and the bowel content is demonstrated by the normalization of this relative deficiency in the function of dendritic cells in the intestine of cirrhotic rats with ascites after bowel decontamination. And this relative immune deficiency should be added to the finding of the group from Germany, Reiner and his group, about the deficiency that exists in cirrhosis in the production of antimicrobial peptides by Paneth cells. This among many other functions, antimicrobial peptides are involved in the control of microbiota. It was demonstrated that rats with cirrhosis and ascites, and mainly those with bacterial translocation, show a reduction in the synthesis and expressions of alpha-5 defensins, which is more pronounced in the ileum and cecum, and is linked to the presence of liver insufficiency, since it was not observed in another experimental model, which is the rat with prehepatic portal hypertension, portal vein ligation. And with the help of and the counseling of the CLIF consortium, we were able to characterize the concept of cirrhosis-associated immune dysfunction as a situation, as an entity that is present in advanced cirrhosis at the systemic level, where coexist systemic inflammation and immunodeficiency. I dare to propose a similar situation at the intestinal level. The coexistence of inflammation and immune deficiency. On the one hand, we have at the intestinal level a reduction, an impairment in the mechanism of innate defense with reduction of antimicrobial peptides, which is a direct consequence of cirrhosis. But we also have an exhaustion, a relative exhaustion, and immune deficiency of intestinal dendritic cells subjected to this persistent antigenic bacterial pressure. And this state of immune deficiency coexists with a state of intestinal inflammation with increased expression and presence in the bowel wall of pro-inflammatory cytokines. And all these players cooperate to further damage the intestinal barrier and promote gut bacterial translocation. And maybe intestinal inflammation could be considered a therapeutic target for, to control gut bacterial translocation in cirrhosis. And this is our speculation that one of the mechanisms by which obeticholic acid, the agonist, the FXR agonist, works. This is in a different experimental model of the data that has just presented. Being is carbon tetrachloride cirrhotic rat is a non-cholestatic model of liver disease. We observe treating these animals that have already developed ascites with obeticholic acid. We observe a reduction in gut bacterial translocation, and our speculation was that it was due to an increase in the antimicrobial peptide angiogenin 1 with a subsequent reduction in the total aerobic bacterial count in the ileum, and a reduction in the inflammation in the bowel. This is just one of the graphics with reduction and interferon gene gamma expression and TNF alpha expression. And the take-home messages would be that liver disease is associated with altered intestinal microbiota. And this altered intestinal microbiota contribute to shape the phenotype and function of intestinal immune system cells. And we should consider that there are abnormalities in the immune system at the bowel level in cirrhosis. There is an impairment of Paneth cell innate defenses, exhaustion of immune system cells, and intestinal inflammation. And intestinal inflammation should therefore consider a therapeutic target to control gut bacterial translocation. And before the end of my talk, I just would like to represent in this slide the question that moderators did previously to I don't remember, the hen and the egg to Jas. Which is first? We do not know. Is first dysbiosis that leads to intestinal inflammation, and tight junction dysfunction would be secondary to the dysbiosis, or maybe cirrhosis leads to tight junction dysfunction, increased intestinal permeability. In other words, which is the factor that triggers the cascade of abnormalities in the intestinal barrier in cirrhosis? And I had one slide of the data that commented you on Jas. The study by the San Diego group, in which in two different models, a cholestatic and a toxic model of cirrhosis, they observed that these changes occur very early in the evolution of cirrhosis. This one is increased intestinal permeability, endotoxinemia, tight junction dysfunction. Dysbiosis occur very early, even 1 day after bile duct ligation, and 24 and also after one injection of carbon tetrachloride. And what is interesting is that bowel decontamination with reduction, these changes seems to be independent of the dysbiosis, since bowel decontamination reduced the colony forming units in the second of bile duct ligated rats, but didn't change increased intestinal permeability and endotoxinemia. I would like to thank all my collaborators that work while I am here, and all the audience for your attention.